Hi there, welcome everybody to the second AGI Young Investigator webinar series. As you know, the Ataxia Global Initiative is hosting this webinar series for young investigators in the ataxia field to introduce you to important aspects of various research topics. The second webinar series of the Ataxia Global Young Investigator Initiative is iScience, about hot topics in the respective field and future research perspective. Today, the AGI Working Group 6, Next Generation Sequencing, Genomics and Platforms, will give the webinar Repeat Expansions in Ataxias, Direct Sequencing Detection by Short Read Whole Exome Genome Sequences versus Long Read Whole Genome Sequences and Novel Forms.
Thank you very much, Heike, for this nice introduction. Or maybe to go back one, Stephan, please, just to say 2 words of introduction. So it's my pleasure to welcome you all also in our name. And actually, I'm just the introductory guy. So it's actually really a collaborative effort we are presenting here. And I'm just the introductory guy. And you're seeing the names on the slides. This presentation is going to be given together from Brent, Fogel, Stephan Zuchner, Matt Danzi, and David Pellerin. So it's really a collaborative effort. And if you're a geneticist working on a daily basis with repeat expansions, this is not going to be your presentation where you should hang on to. You can go have a drink, but don't hang around here. If you are new to the field, if you want to learn about repeats, if you want to get some first ideas how you can sequence, how you cannot pick them up by sequencing, then you're perfectly right for tonight for this presentation. The way how we would like to move forward, just to give you an overview on the outline. Stephan, next slide, please. Looks as follows. I would start off with a little introduction about coding and non-coding repeat expansions in textures and frequencies, just a quick update there. And then we start with the real part, number 2, which is about both conventional as well as latest NGS, which stands for the next generation sequencing methods for genetic sequencing of ataxia repeat expansions. And we're going to highlight both strength on the one hand, but also limitations, how to detect coding, but also non-coding ataxia repeat expansions. And we will have a quite high level, but still, we hope, illustrative overview on the main methods. We will start with, let's say, the most conventional one, start off with the direct repeat sequencing, fragment analysis, then go next with whole exome sequencing, then go to short read whole genome sequencing, and then what is likely the latest way and maybe most advanced way of detecting repeats with long-range whole genome sequencing. After introducing to those 4 levels of methods, we then show the application by means of 2 use cases. And those 2 use cases will be the 2 most recently identified, and not only most recent, but also most frequent ataxia repeat expansions, ARC1 and GAA FJ14. And David Pellerin will show you how they can, but also how they cannot be picked up. So we go from methods to what I think quite catchy and quite nice, interesting late use cases on the ataxia repeat expansions. So let me start with an introduction. Why are we talking a whole webinar just about repeat expansions? If you come from the ALS field or whatever, you said, come on, we have 1, maybe 2 repeat expansions. Well, why do you want to spend the whole evening on just this topic? This is because this is a completely different story in the ataxia field. And let me illustrate this to you just by the mere fact of frequency. If you have a look at the autosomal dominant ataxias, you see an overview of 320 autosomal dominant ataxia patients, a consecutive cohort from Germany, and you see the relative frequencies in this pie chart. And what you can appreciate is just the most frequency. If you see from the upper right part, SCAR3, then already SCAR27B, SCAR6, SCAR1, SCAR2. So just the 5 most common autosomal dominant ataxias are all repeat expansion ataxias. So as a total, they account for more than 70% of all autosomal dominant ataxia patients do have an underlying repeat expansion as the cause. And to my knowledge, there's no other, at least no neurological disease, and I don't think no other disease at all, where such a high, just mere by the pure sheer number of patients do have repeat expansions. And just to illustrate this, why did we pick SCAR27B as a use case? David is going to focus on. You see, this is SCAR27B is very recent, but maybe one of the most common, or maybe the most common autosomal dominant ataxia. So how about the autosomal recessive ataxias? Yeah, it's a little bit more complicated, but what you can still appreciate on the upper right part, FA stands for Friedreich ataxia. The by far most common recessive ataxia is again a repeat expansion ataxia. And you see on the lower left side, Stephan, next please, you see RC1. RC1 is also an ataxia, which means the most common recessive ataxias are repeat expansions, and let's say part of the most common, another one is RC1. So two repeat expansions belong to the 5 most common also recessive ataxias. And the total of, if you aggregate RC1 plus Friedreich ataxia, more than 30% of all autosomal recessive ataxia patients are again being caused by an underlying repeat expansion. And this just adds to the fact by the sheer frequency of ataxia patients being explained by repeat expansions, why this is such a special topic. But it's also not just the sheer number of patients, but also the sheer number of different repeats. Next one, there are more than 20 different ataxia repeat expansions. And again, there's no other disease where there's so many different repeats. And this is a non-exclusive figure, which I'm showing. It's also already from 2019. This is just now going a little bit more deeper into and already bridging to the methods part. What you can see is not only are there more than 20 different repeated expansion diseases, there again, RC1 and SCAR27B are even missing there, but what you can already appreciate also that they are not all of the same type. First of all, some are exonic and others are intronic. And this will come again important if we talk about exome or genome sequencing, being able or not being able to pick them up. So keep this in the mind. Is the repeat expansion intronic or exonic? A second thing to have and keep in mind when looking at repeat expansions, what exactly is the motive of the repeat expansions? You all know CAG, or also called polyglutamine repeat expansions, because they are the most common. They are around there since the 19, SCAR1 to 3, SCAR6, 17. So you all know them. But then already Friedreich ataxia is a non-CAG repeat expansion. It's a JA. And you see on the slide also there are all different types of motifs. And why it is important to pay attention not just to the fact, is it a repeat expansion ataxia or not, but also to the specific motive. This topic will come up again in the last part from David Pellerin, where he will focus and show you example that maybe not just a repeat expansion per se is pathogenic, but also it has to be a certain motive. So this is the second way of looking at the repeat expansion, not just exonic, intronic, but also the type of motive. Those are 2 dimensions to look at this. So how can you sequence this? And with this, I would like to hand over to Brent Fogel, who will introduce you to the first type of methods, how to detect repeat expansions.
Fantastic. Thank you, Matthis. So now we're going to talk about how you would detect all of these different types of repeat expansion. So I'm going to sort of start in sort of a historical type of a way. So initially, one of the first ways that this was done was using a Southern blot analysis, and many can still consider this to be the gold standard. In this method, which is shown up there in the upper left corner of the screen, basically what you do is you take a genomic DNA, you fragment it, you separate it out on a polyacrylamide gel, and then using a probe, typically radioactive, but there's other ways of doing it, you look for the region where the repeat is located. And so what you can see here is in this illustration, you have an example of where a normal allele would fall, and you have an example of where a expanded allele would fall. And excuse me a moment, I have some dogs who are incredibly interested in this topic. Now, there's some problems with this. This is a very, essentially a labor-intensive type of a process, and it's very low throughput. So there's, what other methods can you do that would be a little bit faster and more high throughput? So obviously we can go on to PCR. Now we can go on to standard PCR, where you simply PCR across the repeat region and look at the size of the resulting band. But I'm going to skip over that quickly and talk about repeat primed PCR, which is more common. So in this case, I'm showing a region here that's expanded. You have a flanking PCR primer that sits in a region just outside the repeat, and then you have a primer that sits in the repeat itself. Now, because the repeats are a variable size, the primer can sit anywhere. So what you get is a tracing that looks very similar to what you see up in the upper right-hand corner, where in an expanded section, you get a stuttering effect that goes down the line there on that tracing. And that tells you that the repeat is expanded over the normal length. Now, there's a number of advantages to this technique. It replaces the challenges of the southern blotting. It's inexpensive, it's high throughput. It's more versatile than standard PCR. With this method, and I'm not illustrating this now for the sake of time, you can see sequence interruptions, and also you're not really dependent on the size of the repeat. You can see very large repeat expansions here, whereas with standard repeat, you may have to use different techniques if the repeat is very large. There's still disadvantages to this. It's still a time-intensive process. The results are subject to how good your PCR is. You can't determine the exact sequence. One thing we haven't talked about is some of these sequences can be interrupted, and that may affect the pathogenicity of the repeat itself. You also can't distinguish between heterozygous and biallelic expansions by this method, and that's going to come up when we talk about the recessive repeat expansions like RFC1, where you have to use a second method to show whether or not the expansion is biallelic. And then also size is not directly measured by this method. In southern blotting, it is, but in this case, you're estimating it compared to size standards. So there may be a little bit of inaccuracy between the repeat sizes. Next slide, please. Okay, so what else can we do? So we can talk about direct sequencing. And so one of the ways that people might approach this is simply direct sequencing by Sanger method, which is the simplest thing that can do in the lab. Most laboratories can do this. It's inexpensive. If you could size across, you could accurately size the repeat if you could sequence across it. So I'm illustrating an example here. We've got a repeat expansion. This happens to be a CAG expansion. So triplet polyglutamine. Now, I have some example tracings of the sequencing shown there on the left. So we have a long allele where we have a very long range of these repeats. And then we have the shorter allele where the repeats end after a period of time. Now, when you do this type of sequencing, you can't separate out the alleles unless you happen to have very fortunate and have specific primers that can do that. But most of the cases you can't. So when you merge it, what happens is because of the unequal lengths, you end up getting inaccuracies of the sequence. As you can see, it's as when the two sequences, when the repeat on the longer allele overlaps with the shorter allele, you get a whole range there of ends where it becomes unreadable. So despite all the advantage of this, it's very difficult to use this technique to size or to sequence larger expansions. It again is also very time intensive. You do one at a time. So the only time this is really effective is in very small repeat expansions, maybe something like SCA6, for example. But most of the time, this is not something we would want to do. So next slide, please. Of course, we want to think about what we do routinely in clinical practice, where now we're thinking about next generation sequencing. Now, this is a little bit out of order because initially these methods were developed for whole genome sequencing, and that's really what we're going to hear the bulk of from Stephan and Matt. But I'm just going to introduce this from exome because logically it's the next step up. So in this example, we have this same repeat region. How would we identify this by whole exome sequencing? So now in this case, what happens is the DNA is fragmented. And when the DNA is fragmented, you end up with these pieces that are about 50 to 300 base pair in length. Now you then assemble them and align them to the human genome. Now the problem is because these are short, they don't represent the entire repeat region. So you get regions that overlap each other, and you don't really know how to order them. So we've illustrated that here on the left side. So now you can use computational methods where based on the number of these, you can make an estimation for the repeat size and whether or not it's expanded over what the normal length is. Now, this is really helpful in a lot of ways. It can be done very rapidly, it's just part of the exome sequencing. It can be very high throughput, and you can assess multiple genes at once. Again, this is exome sequencing, so we'd only be thinking about repeats that are in the coding sequence. Again, when we get to genome sequencing, then we can think broader. Now, the problem again is if you want to know the exact number and the exact number of repeats in the exact sequence, you have to be able to overlap this entire fragment length. And so that isn't most of the case. So most of the time, this has to be done bioinformatically. So there is a estimation here of the size. It has to be computationally done. And this may not work for all repeat expansions. We'll hear more about this as we get into whole genome sequencing. So with that, I'm going to move forward to the next slide and turn it over to my colleagues to finish off this discussion.
Super. Thanks, Brent. This was great. So to sort of follow up on this logical way upwards, as you all know, or if you just heard, the best way would really be to use direct sequencing instead of having many different molecular methods. Some of them got quite challenging on a sort of in the lab and hard to scale really. Wouldn't it be great to use next generation sequencing to have a much more efficient method, also to start screening maybe larger samples and genome bites? So, and of course, a big reason is this has become quite affordable and will continue to become more affordable. Also, as you may or may not know, the human genome was actually only fully finished last year with the so-called telomere to telomere genome effort that was published in Science. So only now the genome is fully mapped, and there's a fairly complete now blueprint of the human genome available. And the reason that was not possible before is that about 50% of all the sequence in the human genome are structural changes and repeat structures. And they are very hard to explore with the so-called short read sequencing that Illumina provides. And these two new companies, or not that new, but they provide technologies that are now become very interesting. Oxford Nanopore and PacBio, they really focus on the so-called long read sequencing. Now, what is short and long read sequencing? Let me see. This might not be fully what I wanted to show, but anyhow, the idea is that the so-called short read sequencing that we all use in whole exome sequencing or whole genome sequencing really creates millions and billions of reads that each are about 150 base pairs. And they are mostly paired reads, so they have a sort of a linker where we don't know the sequence information of it. We can have an idea how long that is, and then we have another short read at the end of it. So that's called a paired read. But at the core of it, it's only 150 base pairs of true sequence. And as you can quickly understand, in Huntington's disease, for instance, where the pathogenicity level starts at 40 repeats. So that's 40 times 3 with a CAG repeat. That's 120. So 120 bases, as long as you can read this, you can actually directly sequence this with short read sequencing. It works. Now, in the example of FGF14, where the pathogenicity level starts at somewhere between 250 and 300, certainly 300, you clearly see you way above this threshold. So there is just no way you can directly sequence the FGF14 repeat with short read sequences. Okay, and that is the reason why, even though you see next generation sequencing all around you for 10 years now, arguably there were still relatively few major discoveries. There were some really major ones, but maybe there are a lot more when we truly could sequence actually these things. Now, with this limitation of short read sequencing, I mean, there are many benefits, but the limitation of the short reads and how do you map them so that it's very clear where they belong, where they came from. People have still tried very hard to optimize and improve the situation, and especially in how bioinformaticians sort of align them. That's the process of taking a single short read and deciding where in this long genome belongs this read. And so the software has really improved, and they have used all kinds of tricks to get the maximum out of it. And some of the latest tools are the so-called Expansion Hunter and Expansion Hunter de novo software, or Starling, or a tool called Gangster. And I would argue that probably this generation of software tools might be sort of the top of the mountain, what we will ever see, to extract the last bit of information out of short read sequencing data. And one reason is that there are many generations of other softwares have led to those. They're fairly new. But also the world is definitely turning to the next technology, which is the long-read sequencing, where some of this will simply not be needed anymore at that sort of level of sophistication. So I think I talked already about this. Another thing to consider is, as you know, repeat expansions are not purely about the length. It's also important to understand, are there impurities in these repeats, and that can sometimes lead to that the allele, the repeat expansion, is actually not pathogenic. So the short read, as you can imagine, has additional trouble to evaluate if this is a pure or not pure repeat, but if you can't sequence through the whole thing. So with these softwares today, we can measure expansions to about 150, 170 bases total. We can make an estimation. Yes, there is more material here. This is probably longer than 170 base pair or 150 base pair. But everything becomes more uncertain beyond that point, and it needs these confirmatory studies that Brent talked about, or it needs long read sequencing where you can actually directly see the signal. But still, these tricks have helped. And for instance, FGF14 was discovered, and to our knowledge, probably the first locus where we found it genome-wide without linkage support. In the paper, we talk about families, and they have tremendously helped. Without them, wouldn't have been possible. But the actual linkage was somewhat misleading for different reasons. And Matt Danzi here will talk about long read, but he saw the signal first in a genome-wide short read approach. And we dug into the signal, and then David Pellerin was key here, and we proved that FGF14 is actually a repeat expansion locus. And here's the paper that came out earlier this year. At the same time, another group from Australia found the same locus, which is great because it immediately confirms the finding, cross-confirms it. So fantastic achievement. But the truth is, when long-read genomes become more widespread, more available, this work probably will get a lot easier. And that's just how it goes. You know, as progress happens. We did, however, apologize, the formatting is off. We did use short read in our group to use these tools and get the most out of genome samples, just to understand the general landscape of repeat expansions in the genome, pathogenic and not pathogenic. And generally, you know, what you can see is that there are many more repeat loci in the genome than previously appreciated. That comes out of this new software application. And there are now papers out or about to be published that say that there are 1.7 million and more loci in the human genome that are a tandem repeat. And each one of them could potentially expand and may or may not cause a phenotype. In fact, every single genome appears to contain 250 or so fairly common tandem repeat loci, and also a number of rare ones. And when you look at the population level, this is a cohort of over 2,000 whole genomes. The common variants or tandem repeats loci, those you see again and again when you look at the population. But the rare loci, you find actually kind of, quote, new ones the more genomes you actually study. So that very much mirrors what we know from single nucleotide repeats. It also tells us there is a lot more material out there to work with. And some of those could, in fact, cause human phenotypes. And so this is a quick summary of the advantages and disadvantages of short-read sequencing. And I think I've gone through all of them already. Matt is going to tell us about the future, which has kind of started, but be early into the game of long-read sequencing. Matt.
Absolutely. Thanks, Stephan. So there are two major providers of long-read sequencing technology. I'm sure that number will expand in the very near future, but for now, two major players. The first is Pacific Biosciences, often called PacBio. The second is Oxford Nanopore, or ONT. PacBio's approach for creating these long reads and what exactly they are is at least vaguely reminiscent to the kinds of things you might be used to from Sanger sequencing and Illumina sequencing, in that there's a DNA polymerase and we're looking at what nucleotides are getting incorporated as it goes along that DNA strand. Nanopore's approach is completely different. They have a membrane, and then they have a hole in the membrane filled with a biological nanopore complex. And then they feed a single-stranded piece of DNA through that nanopore and measure the change in voltage across the pore as that piece of DNA goes through. And that's shown with this light blue squiggle on the right side of the screen. And then they interpret that squiggle DNA sequence. So a very different approach. And kb typically, and nanopore in particular can yield very, very long ones that are hundreds of kb, even over 1 million base pairs long is not unusual these days. You may have a basic intuition that as these DNA sequencing measures get longer and longer, are they getting less and less accurate? And generally that's kind of true. Both of these companies have historically had a lot of trouble with producing really accurate DNA sequences, not just really long ones. PacBio, I have diagrammed out their approach that largely solves this problem for them, what they call their high fidelity, the exact same piece of DNA over and over again, and then take an average, and then that's about as good as you get from an Illumina machine, those short read whole genomes. Nanopore has something similar called duplex that they're working on, but are still kind of struggling with this problem of how accurate are their reads. But either way, you're getting things that are 10,000 base pairs and maybe even hundreds, over 100,000 base pairs with these approaches. Next slide, please. When it comes to the question of, okay, so how do you actually analyze repeat expansions with this technology? It's actually really, really simple. If you imagine a stretch of repetitive DNA shown in red here, surrounded by non-repetitive DNA in this person's genome shown in black, your long read data goes all the way through it. It'll align into the left side and the right side, and you can just kind of look at what's happening in between those 2 points. And in general, that's what's going on there, usually within a few base pairs of what Sanger sequencing or other wet lab techniques will tell you. You don't have to go through a lot of the difficulties that you might have with short read, and you're having to make statistical inference about how big this repeat is. And since these long read sequences are 10,000 or more base pairs long, they're easily long enough to do this for not just the short polyglutamine, short-ish polyglutamine repeats, but also the really long ones like FGF14 and RFC1 and anything else that might be several kilobases long. Next slide. As for how to actually resolve the DNA or the DNA sequence of these repeats in any kind of high throughput manner, there are a lot of tools out there that can help do this. Unfortunately, there's no clear best tool yet that works for both PacBio and Nanopore technologies. There's a tool called Target made by PacBio employees. That's probably the best tool out there for PacBio data. There's a tool called Strike that only works on nanopore data. It works directly on that voltage squiggle. It's really slow, but really good, in my opinion, at least. And then there are a few tools like tandem genotypes and Straggler that only work, or that can work on any technology, but are maybe a little bit less accurate than Target or Strike on their respective preferred technologies. But the good news is Target and tandem genotypes are at least very fast. They can often do hundreds of thousands of loci in just a matter of minutes. All right, next slide, Stephan. The advantages and disadvantages of long reads. Well, the advantages are that it's arguably the most rapid and highest throughput of anything we've talked about here. You can actually look at any repeat locus, whether it's intronic or coding. For instance, and as netted topologies, which is something that is kind of beyond the reach of the short read tools. The downside is it's really expensive. This is by far the most expensive thing we've talked about here. You're probably looking at $2,000 plus per sample to do good whole genomes with these. Maybe that number is a little lower now. And it requires a lot of specific computational and bioinformatic expertise, and that expertise might even be different for the different companies, Nanopore and PacBio. So you might need some an array of different collaborators who can help you with this kind of work. And with that, I'll turn it over to David.
Thank you, Matt. Just show my slides. You can all see this. Yep. Okay. So thank you all for this thorough overview of the different methods to detect repeat expansion. So now that we've learned about the different methodologies to resolve these expansion, we're going to illustrate how that applies to two recently described repeat expansion in RFC1 and FGF14. Right. So starting with RFC1. So fairly recently, biallelic non-canonical, and by non-canonical, we mean non-reference, so not the reference motif, AGGG repeat expansion in the second intron of RFC1 have been shown to be a very common cause of the autosomal recessive cerebellar ataxia, neuropathy, and vestibular areflexia syndrome that we abbreviate CANVAS. And if you actually look at the graph at the bottom of the slide here, this 11 copies of quadruple AG repeat is the actual reference sequence, so the actual reference motif. Whereas the initially identified pathogenic motif was this AGGG repeat. So as you can see, that repeat differs in nature from the reference motif. The pathogenic threshold, as we know it now, is probably above 250 of those repeats. But now, practically speaking, how do we apply the different methodologies we talked about to detect RFC1 expansion? And starting with short read sequencing, well, because that repeat is intronic, let's look at whole genome sequencing. So whole genome sequencing, as Stephan mentioned, can give us only limited information about the repeat expansion in RFC1. And that is because the repeat expansion is essentially in all cases very large, ranging in size from a few hundreds of those pentanucleotides to a few thousands. So much larger than the short read length, which is only about 150 base pair. And so you can imagine that there's no way such short read will span the entire length of that expansion. But before we actually get into more details about that, the reference sequence only has 11 copies of that quadruple AG. And so in normal circumstances, in someone with normal alleles, short reads are actually spanning through that normal repeat locus, and you can actually gather some useful information about their presence. And that can actually be a useful tool to tell that someone actually carries one of those normal alleles. Whereas in comparison, in people with biallelic expansion, because the expansion, as I mentioned, is so large, but also very GC rich, what you often see is a drop in coverage at that locus because it's not as well sequenced and it doesn't really align well because, you know, there are many reads made up of that motif, which is not referenced, so it doesn't necessarily map well to that region. So that's one consideration. The second consideration is that these short read tools can actually not accurately size these large expansion for the reasons we mentioned. And you can only gather some limited information on the nature of the repeat configuration that is immediately adjacent to the flanking sequence, where the reads can actually map with confidence because they do not contain that repeat motif. And so, for instance, in this case here, you see that AAGGG, which is that pathogenic motif we talked about. And so you, despite the fact that you cannot size that expansion, you can still get some information about the potential motif, although you don't get full information over the length of the expansion. Now, you may think, well, you know, this is intronic, probably only, I can only probably use whole genome sequencing, but actually a few studies looked at, can we use whole exome sequencing by exploiting the off-target reads, which represent roughly 15 to 20% of the reads generated in whole exome sequencing. And in that particular study by Rafay et al. In 2019, looking at 27 whole exome sequencing, they were able to show that informative reads, so aligning to the locus, were found in 14 whole exome sequencing, so not in all. Meaning that whole exome sequencing is not a tool with which you can necessarily gather information about the nature of the repeat in everybody. But nonetheless, you know, 14 out of 27 at informative read. But again, the maximum read coverage was quite low, so only 2 reads on average mapping to the locus. And you know, the information that you can gather about the sequence of the repeat is only very limited. More recently, Scribe and colleague actually designed probes flanking the repeat locus and integrated that in their target next generation sequencing gene panel to see if by actually enriching that region in a panel, one could get a better idea or could use that gene panel as a first screening tool for biallelic expansion. And what they actually show was 100% concordance rate with the PCR assay. So which meaning it can be potentially a useful tool pending further validation for screening biallelic expansion. But again, the same caveats apply here, where basically biallelic expansion or identify information about the nature of the repeat motif or repeat configuration. But again, this is fairly limited to only the first few repeats immediately adjacent to the flanking sequence. But in all cases, when you do suspect biallelic expansion on short read, you should obtain orthogonal molecular validation using PCR assays, as Brent talked about. And these so-called gold standard methods are used in sequence to confirm first the expansion, then the nature of the expansion. And in some cases, people may turn to Southern blotting to measure with more accuracy the size of the expansion. So the first step is using a flanking PCR of the repeat locus. So in individuals carrying biallelic expansion, because their expansion is so large, the polymerase is not able to actually amplify the locus. And so you end up with no PCR amplification product on that agarose gel here compared to a negative control where you do see a nice bend. But again, this gives you no information on the motif. Step 2 is repeat prime PCR, where you're looking for that sawtooth pattern indicative of an expanded allele. But again, this is motif specific. And so you need to screen for the normal motif, the pathogenic motif. And as you'll see in the next slide, this is getting more and more complicated and complex as long read sequencing is being systematically studied. But again, this gives you not great information on the motif beyond roughly 150 repeats. So you do not get an overarching overview or a comprehensive overview of the nature of the motif throughout the length of the expansion. And lastly, as I said, optionally, Southern blotting can be used to size the expansion, but you have no information about the motif. And as Matt mentioned, long read sequencing, I think, is really changing our understanding of how complex the RFC1 repeat locus is. These are 3 nice recently published studies And actually, Dominic et al. And Scriba et al., by studying cohorts of patients with canvas and canvas spectrum diseases who were negative for biallelic AAGGG expansion, found that actually a number of additional repeat configuration appeared to be pathogenic in these patients. What's quite interesting here is that you can notice that these repeat motifs are different from the reference motif, which is quadruple AG. So they all differ in nature, whereas that reference motif appears to be non-pathogenic, and this one appears to be variably penetrant, depending on its size. So when it's larger, it appears to have some pathogenicity, whereas when it's smaller, it appears not to have. And this study I thought was quite interesting, where they basically systematically applied long-read PacBio sequencing to a number of individuals. And what they showed is actually in individual with an RFC1 repeat expansion, it is often of a very complex configuration with multiple different repeat motifs actually intermingling. And so you realize how complex that locus is and how this may actually complexify the diagnosis. But nonetheless, what we're starting to understand is that it is not really the exact repeat motif that probably matters for the pathogenicity of RFC1 expansion, but rather the size and the GC content. And so you can have these very complex expansion, as one can see here, with different motif intermingling, but yet they are still pathogenic because of their size and because of their high GC content. And so long-read was really instrumental in helping us understand that better. But you can also understand this will have implication in the day-to-day diagnosis, whereas initially when RFC1 expansion were discovered, we had this rather simple two-step protocol where you would do a flanking PCR, as I showed you, then a repeat prime PCR, and then that would basically confirm the diagnosis. Although with all these new pathogenic motifs, it is foreseeable that in the near future, long-read sequencing will get integrated in the diagnostic algorithm to actually get a comprehensive overview of the expansion when present. So now switching gear, a little bit to FGF14 repeat expansion. So earlier this year, GA repeat expansion in the first intron of the major isoform of FGF14 was shown to be a common cause of the autosomal dominant spinocerebellar ataxia 27B, which is also known as GA FGF14 ataxia. The graph at the bottom of the slide here depicts the location of that repeat locus. And as you can see, it is located deep in a massive first intron, which is roughly 600,000 base pair long. And as of today, our understanding is that the pathogenic threshold is probably above 250 repeats, although as Stephan mentioned, expansion with a repeat count between 250 and 300 appear to be incompletely penetrant. So similar to RFC1, can we use short-read sequencing to detect FGF14 expansion? Well, the answer is we cannot use whole exome sequencing to detect these deeply intronic FGF14 expansion. And whole genome sequencing actually has a role, or is more useful to rule out the presence of an expansion in individuals with two alleles of less than 50 repeats, which are within, which are spanned basically by a short read. Now, what's quite good is that 80% of the FGF14 alleles have less than 50 repeat. So this can be a useful tool to actually rule out. However, this tool cannot be used to rule in the presence of an expansion. And I'll give you an example very soon. And that is because you cannot accurately size expansion above 50 repeat. And again, echoing what Stephan mentioned, we cannot accurately detect motif impurity, which appear here to be important in FGF14 expansion. So just to give you an example about what I just said, this individual with late onset ataxia was subjected to whole genome sequencing, and a genotyping tool called Expansion Hunter was used, and said that the two alleles were 17 and 97 GA repeats in size. So you'd look at this, you'd say, well, 97 is way below to 150. This individual does not have an FGF14 expansion. However, because that allele is above 50 repeat, just remember you should get molecular confirmation, orthogonal confirmation. And sure enough, when that individual is tested by PCR, you see that the true size of their larger allele is 352 GA repeat and not 97, so way above the pathogenic threshold, and so indeed confirming that this individual does carry an expansion. And so the takeaway message here is really that FGF14 expansion are not accurately sized by short-read genotyping tools and need orthogonal molecular validation. And for that, earlier this year, there was a optimized diagnostic or testing strategy published using essentially the same step as are being used for RFC1, where the first step is doing a fragment length analysis of long-read PCR product to size the allele. The second step is to do bidirectional repeat prime PCR, so basically to query the entire length of the allele from both sides. And in most cases, this is actually sufficient to confirm the GA nature and the size of the expansion. But in some circumstances, especially for very large allele or allele that seem to be interrupted or non-pure, you may need to do things like agarose gel electrophoresis or Sanger sequencing, which are a little more time-consuming. And long-read sequencing, again here, can reasonably accurately size FGF14 expansion and identify complex motif and interruption because it spans the entire length of it. And so, potentially in the future, if validated for clinical use, this could become the method of choice for diagnosis. And lastly, just a word about these non-GA pure expansions. So if you recall in RFC1, we mentioned that the non-canonical or non-reference motif appear to be pathogenic, whereas this is the opposite in FGF14, where so far only GA repeat expansion appeared to be pathogenic for ataxia, whereas the non-GA pure, such as GA-GGA, for instance, or these GA-GCA motif appear not to be pathogenic. And this is based from a number of studies that have shown that these expansion, despite being quite large, tend not to segregate with disease now in multiple families, and so are unlikely pathogenic for ataxia, and they do not appear to be more enriched in patient compared to controls. And so this is why for FGF14, not only is it important to determine the size, but the exact nature of the repeat motif, as this is important for the diagnosis. So one should really gather both pieces of information to make a diagnosis of FGF14 expansion.
Thank you very much, David. And I think that's it. I think we are open to questions.
Yeah. Thank you very much. I would like to encourage everybody to ask questions in the F&A.
Maybe while questions are coming up, so please ask questions to any type, whether it's to a disease, to a certain repeat expansion, or to a certain method. I think this is while we are in a team here, we hope to provide answers to all types of questions. And maybe while people are coming up with questions, maybe I can start. Stephan, I got lost. You showed me, and you showed us that in your New England paper of the SCAR27B repeat expansion, you found it via short read whole genome sequencing. But then you said at the same time, you and David said, you cannot pick it up. Can you just explain this again to me? So what did you pick up? What did you not pick up via short read whole genome sequencing with respect to SCAR27B?
Yes, we did, Matthis. Yes.
But how did you do it? Sorry, I don't get it.
Kidding you. Well, basically, Bernard Bray at McGill spent 20 years of his career to collect these families. These are families with ataxia, negative for any known genes in ataxia. Pretty large families that are also what we call French Canadian in origin. So as it's well known in the French Canadian population, there are certain genetic founder effects. So that allowed to at least consider that some of these families are actually may carry the same pathogenic change, whatever it is. The linkage did produce some interesting loci, but they never yielded a true good candidate. So when Expansion Hunter came out, Matthis here Danzi. He was early in contact with the programmer and author of this software, and we were lucky enough to play with it quite early. And then Bernard thought this would be a great example to share data, short read whole genome data with us, and Matt, we started to learning how to apply these early versions of Expansion Hunter. I mean, by now Expansion Hunter is widely used around the world. And I think Matt can maybe talk more about the details of this, but I think he basically saw a number of candidate loci where Expansion Hunter suggested there might be a signal there. But it wasn't quite that easy, right, Matt?
Yeah, sure. I can talk a bit about this. Sorry, I'm still struggling with my internet quality, so let me know if I'm breaking up. So yeah, basically, I would say the short answer is you can see a lower bound of the repeat size reasonably accurately with short read data. I think the example that David showed in his presentation was excellent for that, where we had this call that was like 17 repeats and 94 repeats, but the reality was like 17 repeats and 350 repeats. I think short read is great for that if you know that's what you're looking for. You're looking for something that's at least 150 or 180 base pairs long or something like that. And you say anything that's looks like it's that size or bigger, I know that's just a lower bound, and I'm going to follow up on it in the wet lab. And that's basically what David had to do. I gave him this candidate site because I was all excited about it. That was the FGF14 one, and hadn't really looked at controls. And he saw that there was a separation between the in PCR and long range PCR between the cases and the controls. But when I started looking at control whole short read genomes, I lost confidence in it because they all looked the same, because all these people with 150 or 200 repeat units look exactly the same as the people with the pathogenic length expansions. And so that can be a really tricky part of analyzing these larger sites with the short read data.
Okay, I thought this was a more basic methodological question. I thought, yeah, just comparing short read to the whole long read whole genome sequencing, this I think was a little more methodological question. Anyway, two long answers. Okay, this there's just a quick answer to a short question. Anna is asking in the chat, could you talk a little bit more about the linkage of SCAR27B, please? So just a short answer to the linkage.
Maybe David, you have some more insight into this prior work.
Yeah, well, I suppose this question refers to what you mentioned, Stephan, about we had no previous, we had attempted to do a linkage analysis on that very large family with almost 20 affected individuals or so, or 15 affected individuals. And it's just that because SCAR27B is a late onset disease or very late onset disease, there were many of the younger individuals that did carry an expansion, but that were pre-symptomatic. And so clinically, they, were unaffected, but that's just because they were too young to express disease. And so that's why we didn't have a linkage locus. But obviously, when we found out about this expansion and we tested the entire family, we reran FGF14, or that most significant linkage locus was in FGF14, confirming, sort of the discovery.
Thank you. The next question is, I think, for Matt Danzi. Does allele dropout also happen on long read sequencing?
Short answer, yes, it does happen. If you're getting 30x coverage, it's relatively rare. There's a particular weakness for GAA repeats in PacBio data that's kind of an issue with the way their whole high fidelity sequence averaging works. But largely, if you're getting more than 12x coverage, it's going to be less than I think less than 3% of alleles, or actually maybe less than 1% of alleles are dropped out. So if you're getting good, more like 30x coverage, it's a very rare thing to encounter.
If there are any more questions, please ask them. Maybe I can go ahead with the next question is, Stephan and Matt, can you give us answers to, let's assume you have an exonic CAG repeat, SCAR1, SCAR2, SCAR3. What's the diagnostic sensitivity and specificity with an exome versus a short read whole genome versus a long read whole genome for a typical CAG exonic repeat expansion? And if you don't have data, make a guess.
Brent, do you know, this is a clinical genetics question. Brent, do you know of any study that...
So the only study I'm aware of that has actual data on this was published around the end of last year, beginning of this year, but using whole genome sequencing, and that was from the UK Biobank data. Now, and that was pretty effective, approaching about 90%. Now, in our experience, we've been using whole exome sequencing and looking at that for some of these coding ones, and it seems to be very effective, but we haven't been able to quantify it yet. But it seems pretty effective. I'd say it's probably on the order of about 80-ish%. The whole genome is better. I guess.
That's great. And Matt, I know we haven't published, but we're currently analyzing 1,000 PacBio genomes, and we have actually looked at this particular question. Matt, can we say anything about this yet?
Yeah, I guess I was gonna say on the short read, I agree. I don't really have data on this, but my expectation based on the work we've done is that the specificity is pretty good. It's more a question of sensitivity, that you end up with missing things a lot more often than you end up seeing things that are probably not true.
Matt, can you give me a number? What do you mean by specificity is quite good? Is it not, are we talking about 70% or 90%? Just give us.
I would think specificity would be above 90%. That it would be rare, but I would even say probably over 95%. Rare to see something that looks pathogenic, that is crossing a threshold that then doesn't pan out. That would be surprising and unusual to me. I would say 90-95%. Sensitivity though, your guess is as good as mine. I'm not sure how much we don't see. Long read, I would imagine, is pretty good, probably above 90% for both sensitivity and specificity. Yeah, that's about as good as I could answer that question, I think.
I'll add on to that and say that depending on how you have the sensitivity of your expansion hunter set, for fully pathogenic, I absolutely agree with what Matt just said. For things that are in the indeterminate or gray zone, you may see normals that look like they're just inside that. And then when you do them by an orthogonal method, you'll see they're just outside that. So what we tend to do is we tend to tone our expansion hunter down a little bit, so we get something that's a little bit lower than what it probably is, but we get less of those iffy kind of calls. This is going to work itself out as people have more and more experience with this.
Heike, do we have 2 more minutes for 2 more questions, or do we have to end?
I think 2 more questions are okay. Last 2 now.
Okay, yep. David, for you, why is the non-GAA repeat in FJ14JA not pathogenic? Interesting typo.
Yeah. It probably has to do with the fact that GA repeats are associated with different secondary DNA structure, such as triplex or sticky DNA, and these non-GA repeats probably do not form these kind of secondary structures, which do not impair transcription, for instance. And so that's probably one of the reasons. There are probably others, such as epigenetic factor, that are driven by the GA expansion. So yeah, and I mean, I think we draw most of what we know about that. That's partly what we've learned in Friedreich about these non-GA as well, which do not appear to be pathogenic, especially that GA-GGA, which was shown not to form secondary DNA structure in Friedreich.
Okay, thank you. Last question again for you, David. In the case of RFC1, first of question, have you seen increased lengths of expansions and offsprings of patients? Question number one. And secondly, is the length of the RFC1 repeat related to severity or onset age?
Well, I know that that Andrea Cortesi has actually worked a lot on that and has actually a paper in the pipeline. But I think the idea is that the size may change with transmission. That's the first thing. And the second thing is there may be a weak correlation between the size of the smaller expansion and some of the metrics of severity, but I don't think it's a very strong correlation. And again, that's probably limited in part by the fact that we cannot really very accurately measure the size of these expansion. You know, we have a broad idea of what size they are, but we don't really get like a specific number, like for Huntington, for instance, where we get 40, 41. It's more in the ballpark range. So that might also explain why we see this sort of weak correlation in RFC1.
Thank you.
Thank you very much. So as we are still over time, thank you very much to all the speakers for the very instructive talks, and thank you everybody for joining.